Okay, so hi everyone again, and thank you very much so far for the great sessions. And our third session, I just want to make sure, Bo and Amir, are you here? Just please unmute yourself and just let me know that you are here.
Hello.
Hello, I'm here.
Ah, great. Okay, very good. So our session will focus on cell-cell communication and cell-cell interaction at the two cell levels and multicellular levels, and how information theory actually helps us to understand cellular interactions and cellular behaviors. So our first speaker is Amir Erez from the Raqqa Institute of Physics, Hebrew University. And the title of his talk is Cell-to-Cell Information at a Feedback-Induced Bifurcation Point. So please, Amir, share your screen, and you have 15 minutes. And I would suggest, please send me your questions at chat, and we will have small discussion after each talk. Thank you.
Right. So, can you see my screen?
Yes.
Great. All right. So, hello, everyone, and thank you for the opportunity to present my work. Yes, so I will be talking about cell-to-cell communication today and biochemical feedback. And what's on the menu is first a brief explanation about sensing and response in a single cell. And then I will talk about information between two interacting cells. And finally, we will learn that there is no free lunch. So for me to tell you about sensing in a single cell, first I'd like to teach you all of immunology standing on one foot. That's a big task, but what you need to know for the rest of this talk is that the immune system is a complex and adapting system which senses and monitors the environment and needs to respond if there is something worth responding to. And specifically, its main task is to decide if something is self, in which case it can be safely ignored, or foreign, in which case it should be neutralized and removed. And this is not an easy task because self and foreign can end up looking quite similar to each other. So as a result, sensing has evolved as a very complex cascade of biochemical reactions with feedbacks. And this is what we have here in the diagram. We have this caricature of a cell, and we have the cell surface receptor. This is responsible for transmitting information from the outside into the cell. And then once a signal is sensed, it undergoes this cascade of biochemical reactions with feedbacks until finally we get to a readout molecule X. And X is a proxy for the signal strength. X goes into the nucleus of the cell and initiates a response program. Now, this is, of course, very simplistic. In reality, it looks something like this. So this is a particular a receptor cascade for a T cell, an immune cell, a T cell. And looking at this as a physicist, my first thought is this hopeless? I mean, am I going to have, is modeling this hopeless, I should say? Am I going to have to measure every single rate and every single chemical species here and all the reactions in order to be able to understand how the cell works, how the single receptor in the cell works? Or maybe there is this coarse-grained emergent behavior that could be got to. And we wanted to focus on this, specifically this self versus foreign, this key life and death decision point. And we wanted to understand and control this decision, and to do that using a coarse-grained view, right? So how would you do that? Well, one way to do that, to try to understand something in a very coarse way, is to start turning knobs and pushing buttons if you have some, right? And seeing what happens. Okay, but these are live cells, so how do you turn their knobs, right? So to do that, we used inhibitory drugs, right? And these are real drugs given by real doctors, usually for cancer. And here's an example of the data that we got, right? So we prepare at time 0 the cells in an active state, and this is this black line here, right? This black curve here. This is a probability distribution of the log of the number of molecules. Here, the readout molecule, the so-called X molecule. And we dose it with the inhibitor, and if you give it a small dose of inhibitor, then nothing much changes, and you still have this bimodal state. And if you give it a very large dose of inhibitor, then things change dramatically, and suddenly from a 2-peak distribution, we get a single peak distribution, for example. Right, and we wanted to understand these dynamics and also the steady states of the situation. I'll tell you a little bit about the steady states. Dynamics is for another talk. All right, so what we ended up doing is developing a coarse-grained approach to this thing. So here is our cell, the caricature that we just discussed. And actually, we can think about this readout molecule X as sitting in this bath with this part of this very complex sensing and signaling cascade with lots of other molecules, right, which we did not measure. And we can say, well, since we don't know what these are, but we do know that something is happening, we could imagine some kind of effective model where X feeds back to itself. It can degrade, and more X could be created, right? And we could think about the effective dynamics for X, and then these effective dynamics would have the shape of how the probability P of n to have n copies of molecule X, right? Its dynamics depend on if I had n minus 1 molecules, I could make one, or if I had n plus 1 molecules, I could degrade one, or if I already had n molecules, then by making or degrading, I stop having n. Right, and this is a very simple master equation that can be solved at steady state analytically, and this is the analytic solution. And this is a very curious relation because on the left-hand side here, we have P of n. And this is a probability distribution. This is empirically observed. You just saw an example or a few examples. We have a lot of examples. We have very many examples of P of n empirically observed. And then on the right-hand side, we have a mathematical expression which contains this feedback, this effective feedback for the effective dynamics, which is, of course, a purely fictitious term, right? This is something we just made up. But the thing is that we can invert the relation and thereby extract f of n directly from the data. And then we've developed a mapping that will take f of n to thermodynamic state variables that tell us something. Okay, so what do we hope to learn? Well, we hope to learn how the system goes smoothly from this bimodal state to a unimodal state and you know, everything in between. And we ended up considering the fact that since things tend at near critical points tend to be universal, maybe this can help us. And we went to sort of the workhorse of statistical physics, the Ising model, which can undergo this bifurcation from an ordered phase to a disordered phase, or a high temperature phase to a low temperature phase, or a bimodal, right, where the order parameter in the Ising cases would be the magnetization of a magnet, right, would be either finite or 0, right? And it turns out that this analogy between the feedback, biochemical feedback system and the Ising model is more than just qualitative. It turns out to be actually, the mapping turns out to be exact at the bifurcation point, at the critical point for the Ising model, for the statistical physics system. And they have exactly the same math if you define the coordinates properly, right? So for that, you have to define the maxima n star, and you have to define the magnetization with this parameter nc. So very briefly, right, you see, if we were to fix theta to be 0, right? So theta is this bifurcation term, right? Positive theta is unimodal, negative theta is bimodal. If we sit exactly at the bifurcation point, we can tune this bias H so that we can produce less molecules or we can produce more molecules, right? And NC is just sort of the center of this flat top. This is sort of a typical number of molecules in the system, and everything can be scaled up and down as we scale NC. This is like a free parameter in the theory. And conversely, we can sit at 0H, and then by bifurcating theta, we can go from the bimodal state to the unimodal state. Right, so using this theory, we could take data from 6 different drugs and many doses, and we could reconfigure those data, those flow cytometry data, and into this bifurcation plot where the symbols are different drugs, and the symbol size is the dose. And so far, I've told you about a single cell and about our theory explaining single cells. And now I want to tell you a little bit about how we put this theory to practice when considering information exchange between 2 cells, right? Because immune cells quite often read off information from each other, right? And we wanted to understand information exchange in our framework. Right, so here is an example of a T cell reading off information from an antigen presenting cell, for example. This is a typical immune scenario, and this is how we chose to model the system. So now we have molecule X, we have a different cell with molecule Y, and they both have these effective dynamics that we just spoke about. Here the reactions are explicit. This is one choice of reactions. One can choose other forms. And now we have this joint distribution of X and Y molecules for the 2 cells, right? And when we consider the joint distribution, we can turn off communication between the cells. So they don't exchange any molecules. And then at negative theta, we have the bimodal state times the bimodal state. We just have this 4-peaked thing. And then at the bifurcation point, at the critical point, we have a flat top times a flat top. So we get a square. And then deep in the unimodal phase, we get a Gaussian times a Gaussian. So we get a circle. But once we turn on communication between the cells, then they start to fluctuate in concert. And we can see that at the negative theta, we have this bimodal to flat and then to unimodal state, but for the 2 cells together, right? And turns out that they do also go critical. The 2-cell system is also critical at this theta equals 0, H equals 0 point. Okay, so far about criticality and mapping these distributions of molecules in the 2 cells to our thermodynamic space. And now using that, we could ask, well, where in this state space do the cells communicate best? And to quantify that, we use the Shannon mutual information. So I believe I don't have to explain it to the crowd here. And when we considered 2 identical cells, so they both have the same H and the same theta, and now we ask, well, where is the highest information between them? Then it's at the critical point. The mutual information is maximized at this critical point, and we start to see a possible issue also. And the issue is that near the critical point, the system undergoes critical slowing down. You can think of it as a dynamical system near its bifurcation point that has critical slowing down, or you can think about the statistical physics version of this, where near an equilibrium critical point, you have critical slowing down, right? Our mapping maps between these 2 scenarios, but in both cases, the system slows down, right? And the system slows down in a way that scales with the number of molecules, this NC, this sliding scale, right? So yes, if we increase the number of communication molecules in the system, right, then we will have more information shared. between the cells logarithmically, and this is not surprising. The thing is that we would have to pay square root of NC, right? So this is not a great deal because, of course, the square root grows much larger, much faster than the log does. Another thing of note is that this information drops very rapidly, right, as we turn on H. This is because H biases production or degradation. So imagine these two cells fluctuating in concert, right? And now we're biasing production, right? So now we lose the communication between them because they're just both producing all the time, right? So this is why the information was lost very quickly if we just bias the production. So far, I've spoken about identical cells, but actually the picture I showed you, the cells were really quite different, right? So we can think of several ways to break the symmetry between the cells. And one way is to have one cell that's biased more to produce less molecules, right? And one cell that's biased to produce more molecules, right? So now we can have these two different biases, and we can ask, well, how is the communication between them? And the answer is that they can communicate very effectively along this manifold of states, right? Where they compensate, right? One cell is more of a producer and the other cell is more of a consumer, but this framework allows them still to maintain high mutual information with each other. We can also consider one cell that's bimodal and the other cell that's very unimodal, right? Very centralized and very polarized. And again, there is this manifold of high information states. Here, the relation is a little bit more complicated. This is the black dots here. And finally, actually, we could define collective coordinates, big H and big T, which are these combinations. This is the general case, right? So what we did is we drew 10,000 different realizations of HX, HY, theta X, theta Y, and we calculated their mutual information, put them on this plot, and you see again that the information peaks at this T equals 0, H equals 0 point, right? So this led us to think, well, maybe there is this enticing possibility to cheat critical slowing down, right? Because now we could be quite far away from the critical point, right? With finite hx, hy, theta x, and theta y, right? So potentially, maybe we're not going to suffer from significant critical slowing down because we're far away from the critical point, but we can still have high information, right? So that's what we sought to find, but you kind of know the answer already. And the answer is if you take those 10,000 points that you just saw and you ask, well, what's the relation between correlation?
You have 1 minute.
Yes, this is my last slide. Thank you. What is the relation between the correlation time and the information in the system? And you see that they very well predict each other, right? So there's no free lunch. You want more information, you're going to have to wait longer to get it in this system. Right, so to summarize, I told you about how the two-cell system undergoes a critical transition and how the mutual information maximized at the critical point, and this trade-off between the time it takes to sense this amount of information and how much information we get from a particular realization of our model, a particular number of molecules, and we can't cheat this trade-off. And in the future, we would be very interested to try to quantify movies, right? Because actually estimating the joint probability distribution is difficult, but estimating correlation time is not that difficult. So with that, I'd like to thank you for your time. Please send me postdocs if you have postdoc candidates. I'm looking for them. And I'd like to thank all these people for this work. Thank you very much.
Thank you very much. So some questions, please send me a message if you have some questions. In the meantime, I have a question, Amir. Can you probably suggest how your model would change if you would move, let's say, from two-body interaction to three-body interaction and then to multiple body interactions? I mean, how the system would change if you talk about multicellular system and critical points in the multicellular system?
Well, thank you for this question. So actually, there is a follow-up manuscript that I didn't discuss at all here in this talk, And in the follow-up manuscript, we do consider a version of this model on a line, right? So this multicellular, so it's a 1D version, right? So it's not very imaginative, I guess, right? One cell, then two cells, then many cells in a line. This was the program. Yes, to cut a long story short, this is the same situation. Whereas, in fact, there we go into even more detail, whereas you can get more information near the critical point. In terms of information rate, it becomes less and less effective to be there, right? So that, you know, it's hard for me to say what this implies biologically, but this is work in progress. We're thinking about that, the biological implications of that, right? But it does appear that at least within our framework, it's not very efficient to be at or very close to the critical point. And if time is an issue, which normally in biological systems it is, I suppose. I'd like just to add one more thing. This NC, this number of molecules, right? You can reduce time also by just limiting the number of molecules, not to be in the 10,000s, for example, but to be in the hundreds, right? If you do that, of course, things get a lot noisier. But of course, root of NC is not so bad, right? Yeah.
Right, but this is the mutual information between two cells still, yet, right?
Yes, yeah, no, but the NC remains the important factor even in the multicellular system. Yeah.
Okay, thank you very much. So maybe one quick question. If not, we will move to Bosan. Okay, so we will move because we are running late a little bit. So Bosan, are you here? Okay, great.
You got a question there.
So, there is a question? Yeah, please.
How does the communication take place? What's being exchanged? It's a very simple question.
Excellent. So this could be in the form of a gap junction. So a molecule that is actually really transported immediately, or it could be a soluble molecule that is diffuses from one cell to the other, so they're not in direct contact. But either case, it's some molecule that is produced and degraded by the cell and can be exchanged.
Okay, thank you very much. So I would like to introduce Bosan from Department of Physics, Oregon State University, who will be speaking about information flow in multicellular systems from one to many. Yeah, please, Bosan.
Okay, thank you. Okay, all right, hello everyone. Thanks for inviting me to the workshop. I'm glad to talk about a project my lab worked together with Professor Asaf Zariski's lab recently. I'm mostly speaking on his behalf because he couldn't attend this workshop for personal reasons. So the topic we're talking about today is information flow in a multicellular system and how the information is propagating from one to many. From that perspective, I think we can view a multicellular system as a device that takes environmental stimuli and generate desirable responses. But if you compare a state-of-the-art man-made system versus a biological multicellular system, there's some very important distinctions. In man-made systems, for instance, a CCD camera, we want to engineer the individual units or pixels as uniform as possible. And we also predetermine the variance between the individual units so that they perform what they wanted to perform. We program ahead of time. While in a multicellular system, there's intrinsic heterogeneities between the cells. They vary from one to the other. Their gene expression levels are different. The noise, the copy number of each proteins is different in each cell at each given time. So there's intrinsic heterogeneities that you cannot avoid at single cell level. But also the variance between the cells are dynamic. They depend on what the stimuli are applying and how how their surroundings are. So these varying between the units are dynamically and stochastically self-organized. What we want to do is to use data science techniques to review the underlying the underlying networks of this dynamic system and see what they contribute to the biological processes. And in particular, we wanted to model the cell as individuals that communicate with each other, but they also have asymmetric influence between one another. And this asymmetry, we will use Granger Causality to quantify in a later time. The system we're studying is endothelial cells. The endothelial cells is arguably the largest multicellular sensor in human body. They form a monolayer that covers the inner surface of our vasculatures. If you fold it out, it's about 5,000 square meters or 50,000 square feet in total area. Consists of billions of cells, endothelial cells, so-called endothelium. They form an interface between the blood and smooth muscle cells, and particularly they are able to communicate. As you can see from the right here, the cells are tightly connected to each other, and there are two types of junctions. One is a mechanical junction called tight junction, and the other is a chemical junction called gap junctions. So they can communicate either ways, either through gap junctions or tight junctions. These endothelial cells, one of the major functions of this endothelial cell is to sense the shear stress from the blood flow, which we take as input. And what they produce is they will sense the shear stress, and they will send information, they will process information and tell the smooth muscle cells that are adjacent to the endothelium whether to dilate or to contract the vessels. This process is termed as vascular tones. So in our experiment, we will culture monolayers of endothelial cells. We will apply very controlled shear stress as input, and we will use intracellular calcium concentration as our readout as output. And we'll use input, and we'll look at the relation between the input and the output. Okay, so as a primer, here's typical experiment. We use a computer-controlled PID loop to control the shear stress we apply to the monolayer of cells. And when you see the green, that means there's intercellular calcium concentration is high. So we apply a shear stress and as a step function, for instance, and you can see, and we look at the dynamics of calcium dynamics of each individual cells. The dynamics here is the time derivative of the calcium concentration within each cell. So you can see in the beginning, there's a lot of variability between the individual cells, but over time, they're starting to synchronize. So this synchronization process is what we're focusing on. As I mentioned earlier, we wanted to review the underlying network between the cells, and we think the cells are not influencing each other symmetrically, but asymmetrically. And to quantify that, we use Granger Causality between the time series of individual cells. So this allows us to construct a directed graph that represents the monolayer of what we study. Particularly, we calculate two types of scores: transmission score, which is the probability of having an outgoing edge. For instance, if there's six neighbors and they only have 4 outgoing edges, then the probability, then the transmission score is 2/3. Sorry. If there is, on the other hand, 4 incoming scores, then similarly we call this receiver score is 2/3. So within a single experiment, you can easily see that there's a lot of heterogeneity, spatial heterogeneity of transmission score and receiver scores. Now, furthermore, we will call those cells with higher transmission score and receiver scores in different, give them different roles in the network. In this case, we calculate the z-score, namely the score of individual cell subtract the mean, and see how that is comparable with the standard deviation of the population of the score. We gave the high z-score for leader for transmission scores as leaders, and high receiver scores as followers. And if the cell has both high scores for leader and follower, and we call them hub cells. For the cells that are barely communicating with each other, we call them individual cells. So this way, we quantify the heterogeneity of the population with the division of labor. Using those division of labors and quantifying the synchronization process, we find there's a very close relation between how the system approach to synchronization and the network structure. Particularly, this number hub to individual ratio, namely the fraction of the hub cells versus the fraction of individual cells, is strongly correlated with how fast the synchronization happens, as well as efficiency of the synchronization. But I won't go into detail of this correlation map. If you're interested, in the end, I can share the link to our manuscript. What I'm going to talk about mostly is what happens if you apply periodic stresses to the monolayer, because this is what they're experiencing in our human body. They experience pulsating flows that are periodic. And so here, I hope you can see the movies here. Here's an experiment movie recording where you can see the flickering of individual cells as we apply a periodic shear stress. The stress is applied at the 2-minute period. And you can see in the beginning, the calcium dynamics has a lot of variations between individual cells, but over time, it's starting to synchronize to the global, to the population average. So we want to see how the network evolves and what contributes to the synchronization process over different cycles. Okay, so first of all, as you can see, we quantify the transmission score and receiver score over time, over different cycles. The movie, hopefully you can see that there's increase of the color showing that the transmission score and receiver score are increasing over different cycles. So it tells you as the monolayer become more synchronized, there's more information flow, namely a higher transmission score and a receiver score overall. And similarly, as we defined earlier, there's division of labor, and you can see here as well in the beginning of the experiment in the small cycle numbers, there's a lot of individual cells, but as time goes on, the hub cells and leader cell and the follower cell are increasing. And we also ask at single cell level. So all of the above are talking about the population level heterogeneity, how the division of labor is changing at the population level. At a single cell level, we also look at from one cycle to the next cycle, what is correlation between their individual cells transmission score and receiver score, and we see an increased correlation of the scores. What that tells us is that there's a memory effect here. The cell that has a higher transmission score tends to have even higher transmission score at a consecutive cycle. The cell that tends to be a receiver, also likely to be a receiver in a consecutive cycle. So there's two things. One is the increased information flow, and the other is the memory effect. Together, they contribute to the dynamic evolution of the division of labors. So even though we're giving the cells a cyclic periodic shear stress stimuli, the system is not a stationary. The system is never driving into stationary. Instead, we see a continuous increase of leader-follower communication cells and a continuous decrease of individual and common cells. So this division of labor allows us to see what is contributing to the synchronization process. And now, with all the information, we want to ask what is the driving the synchronization? is the information flow actually driving the synchronization at all? And particularly, we hypothesize information is actually propagating from local to global scales. That is accompanying with the synchronization of the whole population. And to test this hypothesis, we set up to calculate the Granger causality, or the edge probability, not only between the nearby cells, but also from cell pairs that are far away. In particular, we expect the following scenario. In the left case, if information is confined locally, then you would expect the edge probability, or the influence probability between between cell pairs, will decrease as at a longer distance, right? Because mostly the information is probably still confined locally, so the faraway cell won't sense influence of each other. So you will see a negative correlation between edge probability and distance. If the information has already reached far, you would expect the opposite, because nearby cells, they have already synchronized. So the time series of one cell is not useful to predict the other cell because they're already synchronized. They contain the same amount of, they contain the same amount of dynamics. So the edge probability will increase with distance. So you have a positive correlation between the edge probability and distance. And experiment indeed shows that's the case. At the beginning of the experiment, this very small cycles, we see a negative correlations between the edge probability and topological distance. And this correlation gradually increase until it reaches, 0 at around cycle 6. And once you keep going on, the correlation become positive. Namely, the information there at those time, the system has already been synchronized, so information has propagated far away. Okay, so
actually, 1 minute.
This is my last slide. Thank you. So in summary, I hope I can convey the idea of heterogeneity, memory, and information all contribute to the collective information processing of a multicellular system. And I want to remind you, this is arguably the largest multicellular force sensor in the human body. So if there's anything that communication is doing a role, this is probably where we should find it most. And finally, on behalf of all the research team, I would like to thank the funding agencies that keep our lights light. And with that, I want to stop and thank you for your attention.
Okay, thank you very much, Bo. So any questions? So in the meantime, I have a question. So Bo, I understand pretty well how model works when the information is transmitted through cell-cell junction, right? When cells are very, very close one to another. But how do you think it would be changed if the information between the cells will be transmitted through soluble factors?
Right. It won't be testable in this scenario because we have a flow constantly going on. If there's soluble factors, they will be diluted by the external flow. And in our system, the cells are communicating mostly through gap junctions because we have tested with gap junction inhibitors that all the positive results we've seen are going away. So we're pretty confident the cells are communicating through gap junctions at least in this system. But that being said, endothelials do communicate through soluble factors, particularly ATPs that when you give mechanical stress, they will secrete ATP that can propagate and diffuse through other cells. So it's definitely possible.
You have also a lot of cytokines, chemokines. You have like a lot of molecules which are secreted and by one cell, right? And by another cell. But in this situation, the cells are not close one to another. They are located at certain distances. So
yes,
and now you have like a problem of diffusion and not a direct transmission. So how do you think everything can be modeled in this situation?
I think you're right. So there are two parts of this. First of all, if there's soluble factors, it will produce mostly uniform, directionally isotropic effects. I think that actually simplifies things because it has, we understand how diffusion works. We understand the, how the soluble factor the concentration of soluble factor will decay as function of distance. So those things, in terms of modeling, I think we can estimate how much it contributes.
So it will be probably distance dependent in this case, right?
That's right. That's right. Yeah, as I said, but in this particular system we're studying, those soluble factors are playing a minor role because the flow itself is carrying them away.
Okay, thank you. Any more question? Okay, so thank you very much, Boenamir, and I think Amos wants to make a note.